Many of you were here last week when Joellen Russell made a presentation which I understand had a standing ovation. Just 2 points about Joellen that I want to make today. Joellen, interestingly enough, I got a lot of complaints. I don't know the details that you people look at, but they complained that why was she an associate professor? And I want to tell you all that she's an associate professor because she's really young. But today, Joellen, I wrote a letter to the president with my strongest recommendation for full promotion to professor. By public acclaim, I suspect. Congratulations. And I also want to point out, and I'm not exactly sure if they're here, but the ecosystem of the College of Science operates in ways that we honor amazing faculty members in many ways. And Joellen is a recipient of the Tom Brown Professorship in the College of Science. Tom Brown, let me just say a few things before you clap. Tom Brown, one of the greatest innovators in Tucson, creator, founder of Burr Brown, which was then sold to Texas Instrument. When it was then sold, it created a foundation, the Tom R. Brown Foundation, which is managed by his two daughters and others. The two daughters, I hope, are here, Sarah Smallhouse and Mary Bernal. Are you up there so we can thank you for all you do for Arizona? I hope you are. Okay, so today I want to point out one of the things that we do, and John Pollard will describe it in a minute, Assistant Dean of Honors College and faculty member in biochemistry and chemistry, and he's the individual that connects this program to the teachers, high school and middle school teachers in our community. And John, maybe you can tell us a little bit about it.
Thank you, Joaquin. I think it's interesting to think about the fact that none of this here would be here, the institution, if it wasn't for our students, our undergraduate students and our graduate students. That is the reason, actually, that we even can have this event that we have tonight, because of the students that attend this institution. And I can go one step further to say that those students wouldn't attend or be here if it wasn't for the teachers that they interacted with at the secondary and primary school level. These are the true treasures of our community. And as all of you know, teachers are in a real fight across this country to be recognized for their worth and their value. I've had the distinct pleasure over the past almost decade now with the lecture series to teach a class with local teachers who attend the lecture series and interact with the speaker afterwards, and really work to think about ways to take the content from this lecture and translate that to how they promote thinking in their classrooms. And again, this year I have 12 amazing teachers from the local area who are sitting over here, once again attending the lecture, so I think we should give them a round of applause. And in addition to this, I've had the distinct pleasure of, for the last couple years, of also teaching a course for freshman honor students who also attend and are sitting with these teachers. These students also use the lecture as content to explore the ideas and themes of the lecture series. And one thing I can tell you about honors students here in the UA Honors College, you know how our institution recruits blue chip athletes to come and play for the basketball, football, softball team, etc. These are our blue chip scholars who attend here. We have about 4,000 of them here at the University of Arizona, and these students are the future leaders, scholars, innovators, and I can tell you these students are all constantly searching for certainty, if I can use the catchphrase here. They're amazing students, and those are also sitting over here. Now, after the lecture, for many years, both this group, we would take the speaker back to a room and do a QA session for about 45 minutes. And these QA sessions have been so amazing that this year, with support of the College of Science and the UA Honors College. We have, produced by AZPM, we are now, right afterwards, we take the speaker to the green room back here and we film the QA session. It's put up online with the lectures. So you can now see these students and teachers ask really tough questions right after the lecture. The last 3 QA science sections are posted online, and I'd like to give you a little glimpse of what we've been working on.
You said earlier that your research is primarily based in pre-existing data sets, and considering that you also said that experimental statistics are becoming more common in your field, does that support your position that randomized trials are often the best statistical measure, or do you think it depends on what you're trying to accomplish?
So randomized trials answer the what should I do question really, really well. And that's questions of a lot of practical importance, as opposed to my evolutionary biology questions of what happened, which are of sort of more curiosity sometimes.
How do you operate in your everyday life knowing these things? I mean, that shot of the moon, I was sort of left in a state of shock at the magnitude of the universe, because it kind of gave this impression. Having this knowledge and sort of dwelling in that realm of your research, and then you have every day like you have coffee or something. I mean, how do you bring these two worlds together?
On a day-to-day basis, I don't think about the scale of the things that I'm working on. If we did, I think we would go slightly crazy. I mean, we being like people who work on different parts of the universe. But once in a while, it does hit you that, like, I'm 10 minutes late to something, and I'm stressed out, and you're like, oh my God, like I'm truly a speck of nothing, and I'm just continually, rushing against time and whatever, and it's like I'm on a tiny planet in a vast universe, living for a blink of time, and of course, but that's not how you live. I mean, that's not how you live.
Why is it still difficult to get society to care about climate change or even worry about addressing it?
I actually think it's that old tragedy of the commons, when everybody grazes their sheep on the same commons and nobody really counts who's putting the 57 sheep as opposed to 2 on the commons. That really makes everybody feel like it's nobody's responsibility, because we don't own it. You don't own the atmosphere, you don't own most of the ocean. But I have huge faith that if we could actually verify who is doing the polluting, we could actually change behavior.
Thank you, John. Now it's my pleasure to, let's see, where does this go? Nowhere. Oh, yes. It's my pleasure to introduce Donata Vercelli. When I became Dean of the College of Science, I got a lot of people from the medical school and from the departments of molecular and cellular biology and biochemistry saying I had to meet Donata. She was one of the brightest individuals that we had as faculty members in the College of Medicine. And I did. I don't know if you remember, we went out to Vivace and I had an osobuco with you. I remember that. The conversation was way better than the osobuco. And since then, I've just been following Donata and amazed at all that she does. Donata is a professor of cellular and molecular biology. She is the director of the Asthma and Airways Disease Research Center and the director of the Arizona Center for the Biology of Complex Diseases. She received her MD degree from the University of Florence. Following that, she joined the Children's Hospital at Harvard Medical School as a research fellow. And then she became an assistant professor of pediatrics at Harvard Medical School. We were lucky that she came to us in 1999. She seeks to elucidate the impact of environment, genes, and the development of the pathogenesis of complex diseases, particularly asthma and allergy. For her work, which will be discussed tonight, she was named a co-recipient of the top 10 Clinical Research Achievement Awards and the Herbert Pardis Clinical Research Excellence Award, both from the Clinical Research Forum. In 2018, she was elected the first female Secretary General of the Collegium Internationale Allergicemium, and in 2019, she was awarded the Rebecca Buckley Lectureship from the American Academy of Allergy, Asthma, and Immunology. You're in for a treat tonight. Please welcome Donata Vercelli.
Am I unmuted? You are unmuted, yes. Okay, I am unmuted, I am told. So we can start. And first of all, thank you, Joaquin. Thank you for the invitation. The second in my career here at U of A. It's always intimidating, but also very exciting to speak to such an audience in such a place and to see that we can fill something as large as Centennial Hall with people who want to listen about science. This is fantastic. To you, really. The story I'm going to tell you tonight starts approximately 400 years ago, a little less than 400 years ago, with this gentleman here by the name of Antoni von Leeuwenhoek. This was a very curious, inquisitive scientist who was also an inventor and a thinker, and who was very curious about the world around him, and who wanted to see what the naked eye, could not actually catch. And so he built this little contraption. I'm saying that it is little small because it can sit in the palm of my hand, as a matter of fact. And this is a microscope. This is the microscope he built. And you know, it doesn't look like a microscope, but it actually is, and it works pretty well. That little eye that you see is the lens. That little butterfly is what you use to focus, pretty much like you would do in the much more complicated machines that we have in our laboratories. And when he built this, we are in 1666, so way back when, the first thing he is said to have been looked at was his saliva. He put a little drop of saliva under his microscope and saw these little guys happily swimming away in the saliva. And this is credited as being the first observations of microbes, these were probably bacteria, in history, and he called them animalcules. So this, as you can imagine, spurred the whole series of research in science and in biology. And so let's fast forward a couple of hundred years to the midst of the Austro-Hungarian Empire, a hospital that took care of pregnant women, where this very fierce-looking gentleman by the name of Ignaz Semmelweis was extremely frustrated. He was a physician because of the very high mortality of women who gave birth, who died, like there was no tomorrow, of puerperal fever. And he had this intuition that perhaps something transmitted from one woman to the other could be the cause, something microbial, some germ, as they were called and still are called, sort of colloquially, could be the cause. And he did something very simple but very draconian at the time. He ordered all his staff to wash their hands when going from one patient to the other. And you know what? The prevalence, the incidence of puerperal fever dropped by tenfold, very rapidly. Then, of course, almost immediately after, and overlapping, we have Robert Koch, who is the German scientist who really nailed the relationship between microbes and disease. Not only he discovered the microbes responsible for tuberculosis and cholera and anthrax, but also formulated these postulates that we still think about that establish a formal connection between finding a microbe in an environment or in a patient and the possibility that that microbe is the cause of that disease. And finally, Louis Pasteur, the French scientist, he has done so many things. I'm an immunologist, he's very dear to our heart. But in this context, perhaps his greatest contribution is that he really formalized the whole theory about germs being the cause of disease, as opposed to previously entertained hypotheses such as strange something in the air or spontaneous generation and all these different theories. He formulated the germ theory of disease in a very clear, lucid way. All of this peaked in the 20s now of the 1900s with this gentleman, Alexander Fleming, at the time not yet knighted. Fleming was working on bacteria. He was working with Staphylococcus. He was growing this stuff on a semi-solid broth, but he was a very sloppy experimentalist. He went on vacation, and he left a plate on which he was trying to grow the staphylococci on the bench. Disappeared for 2 weeks, and then came back and saw something like that. And his genius was not to be sloppy, but to understand the consequence of his sloppiness. What he saw was, in fact, a series of those dots are colonies of Staphylococcus that grew exactly like where they were supposed to. But then, at the top, is this intruder, a big mold that wasn't supposed to be there, but which, however, had the very unexpected and interesting consequence he didn't know it was a consequence at the time, but was surrounded by an area of the plate that was virtually empty of microbes. So the microbes either were killed or at least couldn't grow. And this is his genius. He understood that there could be a causal connection between what this colony of penicillium might be producing and this inhibition of bacterial growth. And this is how the history of penicillin began and ushered in the whole antibiotic era that we are still in very many ways. And you know, this has been one of the great triumphs of medicine, and it's important to state this clearly at the beginning of a talk like the one I'm going to give you. The mortality dropped very rapidly because of hygiene, and then after the use of penicillin. In World War II, in particular, you know, in the previous wars, most soldiers are thought of have died not because of their wounds per se, but because their wounds got infected, and that's what they were dying of. And so penicillin, thanks to penicillin, he will come home, and probably there was a lot of truth to that. So here is Sir Alexander Fleming now bowing in front of the King of Sweden and receiving his Nobel Prize for medicine or physiology in 1945. So all of this contributed to creating this very entrenched notion that microbes, and bacteria in particular, but not just bacteria, are our foes, our enemies. They have to be eradicated, they have to be eliminated, they have to be avoided in every possible way. And again, don't get me wrong, in many cases this is still true. Antibiotics still can save lives when they are properly used, and certainly there are infections that need to be thus treated. The question is whether this statement that microbes are our foes is always and invariably true. And that's what we're going to be discussing today. And to discuss this, I'm going to take you on a trip. And this trip is going to take us among farms and cows, and we're going to be dealing with asthma in children as a disease. And by the way, asthma is a very serious disease that particularly affects children and adults in developed countries, and that, according to the Center for Disease Control, imposes upon societies a burden that is higher than that imposed by tuberculosis and HIV/AIDS combined. So not a trivial societal burden. The first hero in my talk is this smiling lady, Professor Erika von Mutzius, a very distinguished epidemiologist from Munich, Germany, a very close collaborator and a fantastic friend, who has been studying asthma in children in places such as that. That place is Jachenau. It's a very small village in the Bavarian Alps, and it's a farming village, because indeed that house that you see on the left is actually a farm. It's a single-family farm right in the midst of the village. The family lives there. You have the kitchen on the ground floor. You open the door in the back of the kitchen, and you walk into a stable that has approximately 10, 15, tops 20 cows, so dairy farms. And Erika was studying in the prevalence of asthma in these villages. Oh, by the way, and that wooden structure that you see immediately adjacent to the house is a barn where they keep the hay that is used as fodder during the winter, the long winters in the Bavarian Alps, as you can imagine. And when she started studying asthma in these populations, Erika made an observation that was at the beginning stunning, but then was repeated over and over again. And it was that the children who were born and raised in farms, such as the ones that I pointed to, had a prevalence of asthma that was between 4 and 5-fold lower than the children who lived not in the city, because that could be even you could start thinking about many pollution, other things. No, children who were born and raised in homes such as the ones that you see on the right. So in the same village, but they could be the homes of the baker, or the home of the vicar, or the home of, well, in Bavaria, probably not a vicar, probably a watchmaker or the grocery store. So in other words, in a situation in which the environment, in terms of at least the village, was one and the same, this very stunning difference. And they did a very good job trying to dissect where this signals, protective signal, was coming from, and they found that, in fact, what really protected these children was having contact with cows, particularly with cows, and hay, so the fodder for the cows, and drinking the milk coming from these cows very early in life. This was the constant theme. All of this has to happen in early life, and this is why Eric and I came together, because again, as an immunologist, what this was telling us is that this has to happen at a time when the immune system is programmed. And if it happens at this time, protection was almost complete. But their curiosity went further, and so they decided that, well, maybe let's look at something else. Cows are notoriously animals that have lots of microbes, even before, even at that time, that was very clear. By the way, when you go to places like Jachenau, you can smell the village before you get there, because there are these heaps of dung everywhere. These are farming villages, and it's actually a good smell, I have to say. And what they did is that they measured and correlated the prevalence of allergy and asthma with the amount of endotoxin they could measure in the dust of the mattresses on which these children slept. And what is endotoxin? Endotoxin is nothing but a component of the microbial bacterial wall. So a very non-specific target, but something that was used as a measure of bacterial load. And when they did this, they invariably found a relationship like the one that you see there. Namely, that whenever there is a very high amount of microbes in the environment, the level of asthma and allergy in the children is extremely low. So this inverse relationship, very powerful. This is what came to be known as the farm effect, namely the fact that if you grow up on a farm or in an environment very rich, as an environment very rich in microbes, then children are protected from asthma and allergies. And when Eric and I were asked to write a review on this topic by this notoriously hard-nosed journal, we found no less than 45 different reports which invariably and consistently reported effects of this kind. Once you know what to look for, you find it. As long as the environments, the farm environments that people were looking at were of this kind, i.e. these very traditional farms and the children coming in very close contact with these animals. But of course there were questions, lingering questions. First of all, okay, this is happening in small villages in the Alps, in Bavaria and Austria. Is it really relevant to us? How does it happen? And then there was a lingering question that perhaps was a little more specialized, but goes at the heart of what Joanna discussed in the very first of the lectures of this series. What was the right control? Because, you were comparing these children to other children, but there were, and our friends themselves, collaborators, were very aware of that. You were comparing children that grew up in homes that, yes, were seemingly the same, but having animals there had so many consequences. So what was the right control? So this is where the situation was more or less in 2008, which is the year in which I went. This is going to be a very personal account. I went to a meeting, an immunology meeting in Philadelphia in March, to give a talk of nothing else but the farm effect. And at the end of my talk, I was approached by this gentleman. This is the second hero of our story, Mark Holbreich, who introduced himself as a pediatric allergist from Indianapolis, Indiana. And he told me, you know, my wife, who is a hematologist, and I work with the Amish in Indiana, who have some problems with hemophilia and everything else. So we go to the Amish on a regular basis. We work with them pro bono. We love them, they love us, we trust them, they trust us, we take care of them in very many ways. We have developed a relationship with this community. So I thought, why don't I open pro bono an allergy clinic, an asthma clinic for their children? And I have done that, but no one ever comes. And so he is a curious man. He trained a little bit at National Jewish. He's an inquisitive guy, although he's a practicing physician, but with a very, very inquisitive mind. So he said, you know, I asked myself, could it be that this is perhaps an example of the farm effect? And so he came to ask me. And of course, I was transfixed when I heard this. And so the first thing I did, I tried to get some information about the Amish of Indiana that I knew close to nothing about, and discovered that the Amish in Indiana are a population of approximately 200,000 people who emigrated to the North American continent between the 1700s and 1800s to run away from religious persecution because they are Anabaptists, so German-speaking Anabaptists. And they are coming from an area around, or at least this in Indiana, around Zurich in Switzerland. The Amish are farmers, of course, very traditional farmers. They use no machinery, nothing electric, nothing mechanic to plow the fields. They still use this train of horses. They are magnificent to see, I have to say. They have the buggies that you all have seen in movies and pictures. And they have very large families, on average 8 children per family. They have a very heavy German diet, salt, meat, lots of it. They don't smoke by and large. They don't have no internet. They have no indoor pets. They vaccinate their children by and large. So when I heard this, at the beginning, I wasn't sure what to think. So the first thing that I did, I hooked up these 2 people. I said, okay, wait a second, let's have a coffee, the 3 of us. And so we did. And promptly, Eric understood, of course, that this was a very interesting idea and a very interesting possibility. And so next thing that happens, she and Mark go to Amish country, and they very rigorously assessed allergy prevalence objectively, not by hearsay, but objectively, among these children, and found, in fact, this extremely low prevalence of allergy and asthma among these children, which they published. And at this point, this is when things became even more interesting, because now it's time to introduce the third hero of this talk, who actually is sitting there to my great delight, my friend and collaborator, Carol Ober. And Carol is a professor of genetics, human genetics at the University of Chicago. But she is also the investigator who, for the past 20 plus years, has been studying, or had been studying, the Hutterites. And who are the Hutterites? The Hutterites are also German-speaking Anabaptists, except that they came from an area in South Tyrol instead of Switzerland, but an area that, relative to the area that for which the Amish came, is only 200 miles away. It's almost as close as Phoenix. And they went to several places in Northern America, some of them in South Dakota, some of them in Canada. And the Hutterites have characteristics that, as soon as we started thinking about it collectively, made them the perfect control for our study, for the study we wanted to do in the Amish. Why? Because the lifestyle is extremely similar, very traditional, family size very similar, diet very similar, education, very similar. And on top of that, and this was one of the great perks of having Carol in the group, we could also look at genetic makeup. And it turned out that Amish and Hutterites, they are together in that little circle, are much more similar to one another genetically than they are to any other of the population that we could compare them to. So with all of this in mind, we thought that this was a comparison we could do, we could make. And we wanted to make this comparison. Why? Because, yes, Amish and Hutterites are similar in the respects that I just listed, but they are different in two crucial ways. One is, as I sort of alluded to, the Amish are very traditional farmers. Those pictures we took ourselves, are real life, and it's the way they do farm. They farm the way people used to farm in Europe and in the US 200 years ago, basically. On the other hand, the Hutterites are extremely modern in their way of farming. They use machinery, some of which they themselves have invented, designed. They hold patents. And importantly, this has consequences also in the type of society, in a sense. Because the Amish live in single-family farms. This might very well be the farm where Eric and I stay when we go and visit the Amish. And you have these three buildings, and one is the home, one is the barn, and one is the stable. And you can imagine that the children, the many children in these families, roam the land freely, go to the stables, play with the cows, do all sorts of things. They are totally unrestricted in their access to these animals, even when they are very small. Unlike the Hutterites live in colonies, a little more communally. Every family has a house, but they eat all together, and so they have many more shared activities. And most importantly, all the animals of the colony are kept together in a very big barn. And because of that, or stable, and because of that, if I had to point to this could be the stable in an Amish farm, but if I had to point to the stable in a Hutterite farm, it would be probably out of the screen, far away. And the children are not really, therefore, neither allowed nor happen to get in contact with the animals because the animals are far away, and because there are so many sometimes in these stables that the place is perceived as being potentially dangerous for them. So the question was, does this make a difference in terms of their asthma prevalence? This is where I'm going with this. And we think, and we know now, that the answer is yes. Because look, when we measured asthma and allergy prevalence in these two populations that are very similar for lifestyle, diet, genetics, and all of that, nevertheless, the prevalence of asthma in the Amish was between 4 and 5 times lower than it was in the Hutterites. So there we had it. Not only, but these differences were paralleled and probably caused, as you will see, by a difference in their environment, in the sense that we went back to measuring this microbial load using the endotoxin as a proxy for microbes, this constituent of the microbial wall, and it was between 6 and 7-fold higher in the dust of an Amish home than it was in the dust of a Hutterite home. So this emboldened us, especially me, I have to say, to do the experiment that I had wanted to do for a while, and we did it. We tried to recapitulate the environment in a tube and then in a mouse. We call them electrostatic dust collectors. These are Swifters. They're the ones that you use to clean your homes. And that's exactly what they are. You put them on top of a cupboard. We left them there for a month. Then we extracted the dust, and either from Amish homes or Hutterite homes, and we instilled this dust, either Amish or Hutterite, in the nose of mice that we had also treated with allergens to induce in these mice an inflammation in the airways that resembles as much as possible what you have in asthmatic patients at baseline and then during an asthma attack. And let me just show you one result of the many that we got. We measure the resistance of the airways. And the resistance of the airways increases if you give these animals a bronchoconstrictor, because, of course, the airways tighten, and so the resistance to the flow of air increases. And increases, and this is what happens in an animal that has received nothing except the bronchoconstrictor. If you induce inflammation in these animals with an allergen, of course, the airways are even more constricted. That air has even more trouble going through. So then the question was, what happens if you give also this dust? Well, and what happens is that if we gave the Amish dust, we protected these mice very, very, very significantly against this increase in resistance. If we gave them Hutterite dust, not only we did not protect them, but we made things worse. This study landed on the first page of the New York Times. And it is quite an experience, I can assure you. I would say to our surprise, in a sense, certainly to my surprise, because I'm a bit of a grumpy scientist. I would have never thought that in my lifetime I would have published a paper in New England Journal of Medicine, impact factor 75, truly, and landed with an interview on the front page of the New York Times. And I asked myself, why did this happen? And I think the answer is twofold. One is that we showed with data from here, from the US, not from some remote village in Bavaria, that bugs can be very good for your health. And that's not trivial. And the other, I think, is how we showed it. Is the fact that we had this incredibly powerful comparison of two populations with good controls, and on top of that, we had the mouse data where we have a model that we can manipulate and we can get the data. So I think that the impact was a mix of the significance of the data and how they were obtained. So we were, as you can imagine, delighted. But the question was, what is this telling us about our relationship with the microbes in ourselves and out of ourselves? And to answer this, I need to take a little bit of a detour and take you with me in the midst of what is now happening, which is nothing less than a revolution. And people have called it the microbiome revolution, where by microbiome you mean all the genes in the microbes that form this entity in the microbes. And this is really a tectonic shift in the way we think about microbes that is happening and has been happening for no more than 10 years. We are right still in the midst of it. See, the point is that until, I would say 10, 15 years ago, the tools that microbiologists used to study microbes were not so different from the ones that Koch and Pasteur and Fleming used. They were culturing things. They were trying to culture microbes to then isolate them and study their properties. Until someone came up with a puzzle that became known as the Great Plate Count Anomaly. And the Great Plate Count Anomaly, which is, I think, a great name, is the following. If you have a mix of bacteria in a tube, and you look at them under a microscope, hopefully a little more modern than the one that Leeuwenhoek built, but essentially that allows you to see, you see millions of these things. But when you go and try to culture them, you never culture more than 1-2% at most, at best, on a good day, of what you see with your eye under a microscope. So the question was why? And so as long as the detection of microbes was completely dependent on culturing, we really couldn't understand very much of what was going on. But then something happened, and what happened, of course, were several things. One was that someone realized that there is a particular gene, doesn't matter the name, is the 16S ribosomal RNA gene, but this is a gene in microbes, in bacteria in particular, that has the peculiar characteristic of having both very variable regions interspersed with regions that are very constant. And so the constant regions are what they are in all microbes, essentially in all bacteria, but the variable regions are different depending on the order, phylum, genus, species, and so on of that particular bacterium, that particular microbe. And so the idea now, combined with the power of sequencing technology that has exploded in the meantime, the power of genomics, was and is that we can take advantage of these constant regions that are known to sequence the variable regions that are unknown. We use them as anchors for the tools, the primers, that we need to attach to these pieces of the genome of the bacteria, of the DNA. And when you do that, all of a sudden, we have been flooded with information about the profile, the content, the composition of microbial communities which we cannot see, and certainly we cannot culture. And in doing that, then we have also doubled up, and we have also now developed other tools that, depending on what you look at, can tell you not only what is there, which bugs are there, but what are they doing, what are they producing, what are they releasing, which metabolites are they sending around. So depending on what you focus on. So this combination of very advanced microbiology and very advanced genomics, what has that taught us? A huge amount of things. First of all, that microbes are everywhere. They are in soil, in their airways, in our poop, with all due respect, on the stage, on doorknobs, everywhere. And they are everywhere in numbers that are staggering, and that, in fact, I don't think we can even compute very easily. If the stars in the universe that Ferial was talking to us about a few weeks ago are in the order of 10 to the 22nd, we're talking about cellular microbes on Earth to the tune of 10 to the 30th, which is a number I cannot even imagine. And you know what? We are more microbes than human. In other words, each body contains at least 10, if not 100 times more microbial cells than human cells. We are outnumbered. It's true. It's absolutely true. And so the more current and way of talking about humans now, and note that that's The Economist. This is not a scientific journal. This is how pervasive these ideas have become very rapidly, is that we are superorganisms because our functions are not just the result of what our cells do, but of the symbiosis between our cells and this overwhelming number of microbes that we host. And the key word here is diversity. Diversity, diversity. These microbes are incredible in their diversity. So the microbes on the palm of my hand are not the same that I have on top of my hand, and are not the same microbes that I have in this part of my arm or on this, in fact, ailing shoulder of mine that I cannot move. And my microbes are not the same as your microbes. And this is so true, that microbes are now becoming a great focus of interest on the part of forensics. Because in fact, think about it for a moment. Forensics, if you want to establish who has been where, when, if you look at human markers, you have 25,000 genes, but these are trillions of genes that you can type. So this is happening slowly but surely. There is a sort of appropriation of these type of studies on the part of forensics. We know that microbes are influenced by geography, by diet, by travel. If I go to China, my microbiome in my gut changes because of the diet and because of the travel and everything else, and then reverts back to where it was when I return home. We know that migrations are critical, and there are studies now done very rigorously that demonstrate that it may take a generation, but when the whole population goes in a migration to a different place, slowly but surely this population acquires characteristics that are in terms of their gut microbiota particularly, that are the ones of the place they go to, for better and for worse, usually. And why do we have all of this? What is it doing? I mean, is this simply, are we ignoring one another or are we working together? No. These microbes are actually working for us because they are synthesizing vitamins, they are synthesizing hormones, they are synthesizing neurotransmitters. They are critical for us, for our ability to digest food. We could not digest food without microbes. They are critical for the way we metabolize drugs that we take. They are critical to allow our immune system to develop, and we'll get back to this notion in a minute. Basically, there is very little that happens without input from these guys. And of course, there are associations with disease. And those are striking and puzzling. So we know that, for instance, the gut microbiota and microbes of already at 3 months of age, as early as 3 months of age, are different in children who will or will not become asthmatic by age 4. So there seems to be a trajectory there. We know that in individuals with inflammatory bowel disease, aka Crohn's disease, the microbes in the gut of these patients are very different from the microbes in the gut of healthy individuals. We know that microbes have a lot to do with our blood pressure. These are very recent data that show that the way we handle salt, which, as you know, is a major determinant of blood pressure, is due to how specific classes of microbes in our gut handle salt. And if those get depleted, then you have an inflammatory state that leads to hypertension. And we know that microbes have a lot to do with our mood, even. There was a conference in Amsterdam not more than a month ago, exactly on these themes, and you may hear something along these lines or about this from Kathy next week. So you could say, okay, all this is nice and dandy and flashy, but these are associations. In other words, who does what to whom? Is this a cause or is this an effect? What's cause and what's effect? Which is why several of us tried to be a little more causal in their experiment. And so now I'm going to take you to a lab that exists only in some institutions. We are fortunate enough to have one such place at the University of Arizona. And this is a lab that specializes in keeping, manipulating, and hosting and using germ-free mice. These are mice that are born under absolutely germ-free conditions. They have no microbes in themselves. And it looks like the people who work there look like Martians, because see how they work. I mean, this is a picture that was given to me by our collaborator Karuna Patil. Thank you, Karuna, for that. It's not that you can walk and take a look. I mean, this is like I don't know. It's really a super controlled environment, because of course nothing can interfere with the sterility of these animals. Now, these animals per se are not particularly happy, but they are kept away from pathogens, so they have their lives. Of course, their immune system is not particularly good. They are prone to certain diseases, but under the conditions in which they are kept, these mice are okay. But they are the perfect recipients of microbes. So they are empty spaces that can be populated with microbes, and they can be populated with human microbes. So these mice, in other words, can be humanized with microbes taken from the gut of humans. And then you can ask, what do these microbes do? Because now you're putting them there, so now you are going towards causality. And in doing these experiments, the results are stunning. Because if you take gut microbes, stools, from twins, one of whom is lean and the other is obese, and you give these stools to germ-free mice, surprise, surprise, the germ-free mice that get the stool of the obese twin become obese, and the germ-free mice no longer germ-free, they get the stools on the lean mouse remain lean. So you are programming the metabolic phenotype, the metabolism of these animals using these tools. Cancer treatment. We were all very excited when, in October, Jim Allison and Tasuku Honjo received the Nobel Prize for medicine or physiology for their discovery of immune checkpoint inhibitors, because these are now the antibodies to immune checkpoint inhibitors are used in cancer therapy, often, not always, with very good results. Well, I said often, not always. So if you take gut microbiota from individuals who have responded very well to anti-cancer treatment with these agents, you give these tools to a mouse and you induce a tumor in these animals resist these tumors very well. But if you give to germ-free mice stools from an individual who has been treated but hasn't responded, the mouse will not respond. So even this aspect of the response seems to have a lot to do with gut microbiota. So how could we resist the temptation to ask whether gut microbiota have anything to do with protection in Amish children from asthma? So we used germ-free mice. And again, I'm showing you what happens in the lungs of these mice to the resistance of the airways of these mice, depending on whether, this is the baseline, depending on whether the mice that are made to have inflammation because we give them an allergen receive stools, poop, as we call it in the lab, poop from an Amish child or a Hutterite child. So the animals per se, the germ-free mice, as I said, are prone to these very strong reactions. They don't have a very well-controlled immune system because they don't have microbes. If we turn them into Hutterites, giving them this Hutterite poop, not much happens. In fact, they basically stay where they are, so there is no protection. If we give them poop from Amish children, they are protected completely. So something in the gut of these children is protective. So what do we go with this? I have told you that you can find protective effects coming from microbes in the gut, but I told you earlier in the talk that you can also find protection in the environment from environmental microbes. So are these two independent effects? Are these effects that somehow concur and converge? The model we're working on and what we think is happening is something like this. We know that the microbes in our gut take approximately a year to develop fully. You start out with a very small inoculum that comes from your mother, and these are what is called pioneer taxa, pioneer microbes. Pioneer, because they get to an empty niche that they have to fill. But they don't fill it by themselves. They fill it expanding, but also receiving constant input from the environment. So the gut is constantly seeded from the environment. And this is probably the reason why the environment is so important. And if this process occurs as it should, then you have, within the first year or so, a very efficient diversification of gut microbiota, and everything goes well, so to speak. So if you are in a low-risk environment, you have lots of microbes, a big microbial load in the environment. The seeding into the gut occurs with the right pace, and you develop a very diverse set of microbes in your gut that produce protective metabolites, and so the risk of asthma is low. Which is not the case in high-risk environments where the load is limited, where the diversity of what is in the environment is reduced, and so this whole process is delayed, sort of struggles to occur, and because it struggles to occur and doesn't occur within the right time frame, the risk of asthma goes up. This is the model we're working on. If I am invited back to talk to you, maybe I will tell you how this story is progressing in a few years. But I think that we can conclude saying two things. One is that we know enough already to know that perhaps it's fair to say that some of our best friends are indeed germs. Or, in the words of another friend, a great guy, one of the first who really saw this field and gave birth to this field, we are, fortunately, not alone. Thank you very much for your attention.